Thank you very much. I will be very short, and to start with, I'm pleased to be here on behalf of Mr. Ren Wang, the Deputy, the Assistant Director General, who's supposed to be here to chair this seminar. I'm happy to be here, and I'm also happy to see that the network, the technical network of food safety has become one of the most active network actually in our department. So I can see the participation of people here, but also the participation of people that we don't see that are going to be watching this. And I'm pleased and honored to welcome our speaker, which I don't know, but I'm meeting just now. And the subject also is very exciting. And as was laid out in the abstract, we all know that plant selection and even breeding is done by farmers and has been there for centuries since mankind is here on the earth. And that is, if you have people that did basic genetics, this is really the theory of evolution with mass selection and all this. But now, in the modern world, we talk about biotechnology, where modification is now requires some interventions. And this, you know, that has generated a lot of debate and concern about the safety of food that derive from biotechnology, and also the potential risk that is associated with that. We have here, I'm happy to see that science is behind the evaluation of this risk. And a lot of questions are still there about the technical, the scientific methodologies that are used. And we are blessed today to have Adjunct Professor Bartholomaeus. Okay. Sorry for the pronunciation.
Okay.
That is coming today to enlighten us about all these issues. So, because we have been a little bit late about this, I will give you the floor, and please welcome to FAO, and thank you very much for joining us.
Thank you.
And you have the floor.
Thank you. Well, look, thank you for your interest. I mean, it's a pleasure to be back here. Been here a few times with JMPR, and previously I chaired a nanotech in food and agriculture meeting. So, you know, I'm a little bit familiar with the place. I still can't find anywhere in this building. But, so it feels like I've come back home in a way. So a little bit about me. For the last 20 years I've been a toxicologist, and I've worked in areas in human pharmaceuticals, industrial chemicals, complementary medicines, agricultural pesticides, veterinary drugs. And in the last 4 years of my career, before I retired to start my own consultancy, I ran the risk assessment branch for the Australian and New Zealand Food Authority. So I have a very broad perspective on food safety. Okay? Now, in terms of nanotechnology, biotechnology, food irradiation, as a regulatory scientist, what I'm looking for is, what is the point of novelty? What is it about this thing that requires a different approach to everything else we've done? And there's a lot of parallels, whether you're looking at nanotechnology, food irradiation, or GM. When it first comes in, there's all this enthusiasm, and then people say, well, you know, what if? Maybe this could occur, and maybe that could occur. What happens then is we end up with a pile of regulations based on the what if, which kills the technology, because it makes it extraordinarily expensive to run. But as you do more and more work, what you find is a lot of what you think is new has actually been going on naturally in the plant world or in the physical environment forever. So taking the example of food irradiation, when you irradiate food, you do get unintended chemical changes, but they're exactly the same as what occurs when you put it in the microwave or you cook it. But it wasn't until we had all of that data about food irradiation and the consequences of that, and the normal background context against which to compare it, that you could see that it wasn't the issue that people thought it was. Okay? So what I'm going to do here is talk about biotechnology specifically. Right? Now remember, I've been working in the human pharmaceutical side, where biotechnology has been around for a while. And I have seen clinical trials where they inject biotechnology-derived drugs that have small amounts of recombinant DNA in it into the bloodstream of people. and there's like no effect, okay? So a lot of the concerns that I heard 15 years ago when I first started working in the biotech area were a bit tenuous, but sort of plausible, worth listening to 15 years ago. But what I want to try and show you today is that there is so much knowledge that has come through in the last 15 years about the natural background context that completely really changes the debate. And the big problem with biotechnology is it's not the big companies like Monsanto and Dow and Bayer who are affected by the regulations. They're making blockbuster crops that are earning them hundreds of millions of dollars. So if it costs 10 million for a regulatory package, who cares? But if a little university in Indonesia is trying to produce a potato for a poor remote community that isn't subject to late blight, which requires fungicides, which the people generally apply with very poor occupational health and safety techniques, then that 10 million is just too much. So there's an application, and everywhere I travel in Southeast Asia, I've spoken to people in the last 12, 18 months, I've spoken to people from Laos, Cambodia, Vietnam, Burma, Malaysia, Indonesia, Singapore, and all of those countries are looking at niche applications for biotechnology that are largely being blocked by two things: the overwhelming and disproportionate level of regulation, but also the attitudes that come out of Europe which make it, oh gee, if they're that worried about it, maybe there is a problem. And what I found in terms of risk communication, if you set the bar for regulation too high, people say, well, if you need that much regulation, it must be dangerous. It doesn't reassure people that you need a great pile of data. Okay? So do I have a...
Yes.
Okay. Oh, okay, so I just say okay. So I'm going to go through some basic principles of how Australia looks at regulation. So this is sort of Australian-centric or Australian-New Zealand-centric. We'll sort of acknowledge some of the concerns of biotechnologies today, and then I'll talk about this particularly, the relevance of genome plasticity. We know so much about genome plasticity now. Now, I reinforce the fact I'm not a plant biologist or a geneticist, I'm a toxicologist, so I'm looking at this from a high level. So excuse me if many people in this room will know more about some of the detail than I do. We're going to look at where the variability comes from and what does the data tell us we actually need to make a valid conclusion about the safety of a new biotechnology. It doesn't matter whether it's gene editing or recombinant DNA technology. The same sort of principles apply. Next slide, please. All right, so in Australia we actually have a formalised system that requires us to test any new regulation on a cost-benefit basis, right? So if you're going to impose a huge cost, there has to be a tangible benefit that is worth more than the cost you're imparting. Or there has to be such a human health and safety issue that would generally drive a cost benefit anyway, or some other concern that is sufficient that you can say, no, it's not cost beneficial, but there's this other reason that we think justifies it. But you have to be open and transparent about the fact that you're imposing a cost that's greater than the return. So that's rare that people will do that because it opens the agency to scrutiny, of course, and challenge. So for new technologies in general, risk derives from the nature of the application of that technology. Right? What you're achieving. If I create a sonic screwdriver, I'm a fan of Dr. Who, and all it does is turn screws. Who cares if it uses quantum physics? Right? It turns screws. That's what you're concerned about. If I take sugar and I produce nano fairy floss, cotton candy, right? It could have some amazing physicochemical properties, amazing optical properties, amazing structural properties. But if I put it in a cup of hot tea, all I've got is sweet tea. Right? So we need not to get caught up in the hype and the enthusiasm around the technology itself, and actually look at what it's doing and what it actually consists of. So the key thing is to look at the novelty arising from those applications against the context of natural and pre-existing analogues of those outcomes. We have a basic principle too, that the data requirements should address specific sources of risk and viable risk management options. Now what that means is, you don't ask for data that could be very interesting scientifically, but you can't do anything with it. You can't change your label direction. You can't change how much of something can be in something, okay? So an example would be in Europe, they require an assessment of the level of endogenous allergens in a soybean after it's been modified. What are you going to do with that information? What can you do with it? You can't tell people it's safer or more or less safe, because the natural variation in those allergen levels is far higher than any GM can produce. So it has no value. You can't do anything with it. So it's questionable as to whether it's ethical to ask for it. It's not a particularly expensive requirement, I might add, but still, it's an example. And lastly, I think we need to be careful that a precautionary approach is not necessarily precautionary. There are a lot of examples where the precautionary approach is brought into play, but the consequences of blocking or restricting new technologies on the basis of vague, hypothesised harms can result in greater harm than those being avoided. Right? You've got to look at those sort of collateral effects of saying no to something without any information. It's fine to say, in order to go forward, you need to address this, this and this, and be precise about it. That's different. Right? Because you've got specific concerns, but you have to be able, I believe, a regulator needs to be able to identify what whoever is coming to you needs to do to allay those concerns specifically. So the objective overall in terms of regulation is balance, proportionality, pragmatism, what will work and be safe, cost effectiveness, impartiality, and most importantly scientific integrity. And I would argue that most GM regulation around the world, including Australia, no longer has scientific integrity. It is discordant with the huge amount of data we now have. Next slide, please. I mean, you're all familiar with this. I'm sure you've got it on your walls or something. And it's recently been revised upwards, okay? Every 12 years, we add a population equal to China or India. So every single day in 12 years from now, we need to find 3,000 million meals a day extra. Right? If you think of a meal as, half a litre of volume, you make a metre cube out of that. Every year you could go to the moon and halfway back with a stack of that much food. That is not going to happen, right? From, quaint old-fashioned farming techniques. Biotech is not the only solution, not by a long shot, but it's one tool in the box. And if we're going to feed all these people without wars and, border disputes over land and water and so on, we're going to need to find ways to do it on existing land. Because the only other alternative is to start knocking down, nature reserves and environmental parks and stuff in order to grow food, which I don't think is a great idea. So this is an example of where the precautionary approach may be setting us up for a much bigger harm than anything we're postulating today as a harm. Next slide, please. Okay, so what are we doing? We're altering the genome of plants to achieve new traits. Assessing the safety of the new inserted or induced trait is not controversial and requires only an understanding of the trait itself. So in terms of, if I take this gene and it expresses Cry1A, a Bt toxin, there's nothing controversial about that really. We know what that is. We can do a risk assessment. I remember when I was in the pesticides area in Australia, I actually assessed a data package this tall on Bt protein for use to be sprayed on conventional agriculture. It's a mainstay of organic farming techniques, right? And yet everybody's terribly paranoid of this causing holes in their guts because it's in GM at lower levels than you get in organic food that was sprayed with it, which is interesting. The regular debate revolves around unintended, unpredictable consequences of gene insertion or induced random mutation. Now, unintended effects occur every time you breed anything, right? It's the consequences of that, and the degree to which those consequences are unpredictable. Right? So it's not that it's unintended that's the problem, because they are going to occur through every type of breeding. Cell culture causes quite significant genomic changes. Right? But if it's completely unpredictable from a knowledge of the trait itself and the plant, and it's hazardous, well, that would be a concern. So we want to know that we can exclude those. Genome plasticity and the consequences of this plasticity provides the context for addressing these concerns. Next slide, please. Next slide, please. All right, so what are these sort of concerns? De novo production of high-potency protein toxins. Now, if it were actually possible through a minor change, a mutation or shifting genes or cutting the gene off or having two genes in juxtaposition to suddenly produce a Bt toxin, I'd be really worried as a regulator. I mean, that would be deadly, right? Dangerous stuff. But is this plausible from just transgene insertion? Now, as a toxicologist, I know there are 3 separate things that have to occur to take a normal protein and make it toxic. First thing you have to do is change its structure to be resistant to all the digestive processes in the gut. Acid, enzymes, alkali in the bowel. Okay? Very specific structural requirements, very specific range of amino acids that need to be on the surface to ensure that that's true. Okay? All those structural changes are different to the next step, which is the protein, because it's a large molecule, has to be a ligand for an active transport mechanism. Very specific structural requirements. Then it has to be taken up. It has to resist being recognised by the immune system as just foreign material and phagocytised. Very different structural requirements. And then it has to be a ligand for a receptor to produce some toxicity. The likelihood of that occurring, and I'll give you some specific data if you like to support that, randomly through a is as close to 0 as you'll ever get. Right? Scientists don't like saying nothing can ever happen, but it's pretty damn close to impossible. All right? Because a small amount of change has got to cause so many different structural things, some of which may be mutually exclusive. And that's why most toxic proteins, we're not talking about allergens, we're talking about toxicity here. If you think about the natural world, they're all injected. Right, the venoms, to bypass 2 or 3 of those 3 or 4 requirements. Then it's only got to be a receptor for some specific ligand or some blood enzyme or something. Okay, that's why there aren't terribly many toxic proteins that are toxic orally, and they're much less toxic. Even ricin is much less toxic. So ricin is the poison out of castor seeds, is much less toxic orally than it is if it's injected for all those reasons. Okay. So it's implausible. And, 15 years ago when these were first postulated, you'd think, yeah, it's implausible, but, we don't really have enough evidence. That's not true anymore. We can now say it's as close to impossible as you like to get. How about de novo generation of biochemical machinery to produce a toxic secondary metabolite? When you eat a salad, it doesn't matter if it's organic or you've gone into the fields and picked it yourself from pristine countryside, it's full of pesticides. It's full of natural pesticides. All plants produce pesticides. That's what secondary metabolites actually are. They're pesticides to resist microbial attack or fungal attack, all right? Or to stop cows eating it. I'll give you an example of that specifically in a moment. But you can't do it by changing one gene. Right? You can have something really specific, I guess, say cry1A. But otherwise you need to change, you need a biochemical machinery, you need a process, a series of steps. Can you actually do that from random mutations? What does the data say? We'll have a look. Reactivation of dormant pathways. Now, about 15, 20 years, a guy from the FDA said, well, what if these plants have ancient pathways and changing, just shifting the genome a bit could reactivate a pathway for a toxin? That'd be a concern, right? It was tenuous even then because, evolutionarily, unless a pathway is functional, right, it's going to get more and more mutated because there's nothing to maintain that function. There's no selection for functionality. So every generation is going to decrease the capacity of that pathway to function. And it will need greater and greater change every year to reactivate. But actually, there are examples of where you can get reactivation of dormant pathways, but only in a very specific circumstance, and I'll get to that in a moment. Next slide. That's all right. I tend to move when I talk. It's really quite. All right, so just push the forward again. Some of this has got animation in it, so it still comes up. All right, so this was the proposition, and this is the paper from 1992. All right. Next slide, please. All right, so 2 principal sources of risk in plant breeding. The expression of an inserted introduced gene or modification of the expression of an endogenous gene, random genetic variability arising from the process. But variability in nature is the norm, and I'll give you some specific data on that in a moment. Both phenotypic and genotypic variability in food crops is much greater than previously recognised. One of the really positive things about the unnecessary compositional analysis that we require for GM food is that we now know so very much about the variability in normal food. The concept of the food supply of wheat being wheat is complete myth. You know, wheat planted in the same field, from this end of the field to the middle, will have different composition because of the microclimate, or you know, the pest pressure on the outside may be greater than the pest pressure in the middle. And that compositional variation is far greater than you get by inserting a gene. And I'll give you some info on that in a moment. The potential risks arising from variability secondary to, this slide was originally for new biotechnologies, but it's equally true for biotechnologies, is related to the magnitude of that variability in comparison to that occurring naturally. If we've got a new GM crop and we do compositional analysis, what should we compare it against? The nearest isogenetic line? No. Gee whiz, if you did that for conventional food, you'd never import anything. All right? What we're interested in is the composition of this crop materially different to all the other sources of that type of wheat, rice, soybean, because that is determining what safety is. And it's not a nutritional question either, because if you look at the supply of wheat from around the world, there'll be major differences in the levels of amino acid, just depending on the microclimate or the year it was planted, or what the field was like, what pest pressure it had. Next slide, please. So how variable is the phenotype without genoplasticity? How common are toxins in nature and how variable are they? What is their purpose in nature? How common are gene insertions in nature? What are the sources of those genes? What are the consequences of that insertion? How often do point mutations occur and what are the consequences of those? Is there evidence for activation of dormant pathways? Is there evidence for de novo generation of toxins from mutations? This is the data set we're looking for to answer the questions around what is the source of risk really. Next slide, please. What time would you like me to finish? This one, because I could go on forever. 11? No, earlier?
No, later.
Later?
No, 11, so that we can have 30 minutes of
Okay, that's right.
discussion.
I've only got 38 slides, so that should be okay. But I sort of warmed to my topic and I could go. All right, so crop variability. Let's look at some examples. Now, next slide, please. You can go on to the ILSI, I don't know, it's still IFPIC. IFPIC was disbanded, but the ILSI site, there was a compositional workshop done in Washington in 2012, and there's some brilliant slides, and some of the slides I've picked up here have come from people who presented at that meeting, and you can actually get the talk over, so the slides and also the voiceover. So if you're interested in the topic, you can get a lot more information than I've got time for today. Okay. So we're looking for things that could be toxic. Where might we look? Well, let's have a look at what's in nature. This is Patterson's curse, it's called in Australia. Some fool Scots woman thought it looked pretty and brought it to Australia as a nice border around her garden. And now it contaminates massive fields and it causes cancer in cattle and sheep, which is not nice. In humans, we can get, these are pyrrolidazine alkaloids, and you may know that in certainly in some of the Middle Eastern countries, if they have a bad year, in crop year, the weeds are higher and there's more weeds, and then they harvest their grain, they get some of these seeds in, and you get massive ascites and liver failure. But you never get cancer in humans. It's interesting, but I won't get into that because it's another one of my interesting topics. But anyway, so that's a pyrrolidazine alkaloid. Occurs a lot in nature. Quite toxic. Next, can you just push the forward button again for me so I can go back? There should be more on this. Thanks. So, secondary metabolites. This is a secondary metabolite. Why does the plant produce it? Because if a cow eats it, the cow dies. So it tends to discourage cows from eating it. Now, in some parts of Australia, we have such bad droughts, this is actually called Salvation Jane. Because in those parts, even though it'll eventually kill the cattle, it'll keep them alive long enough to give them other food. So whether it's Salvation Jane or Patterson's curse, depends which area in Australia. Alright, okay, it's a herbivore anti-feeding. It's a response to stress of this plant. Next, just push forward, so just get the next lines coming. Okay. These are examples of secondary metabolites. And you can just see there are thousands and thousands. All of these are produced by plants as natural pesticides. And if you stick them in a rate, many of them will cause cancer, and you put them into a genotoxicity assay, they'll cause, genotoxicity. Right? After 20 years as a toxicologist, anybody who says, oh, I like things to be natural, I can tell you I don't, because the natural is more toxic, right, than the synthetic. And boy, that's hard to convince anybody of. But anyway, keep going. There should be some more text here. Alright, so some of these are anti-feedants and endogenous pesticides. They can also be, so we're looking for potential candidates for changes that might be of interest. Changes in lipid profile, mineral and vitamins, heavy metal uptake. So we know you plant potato in a field with a lot of cadmium. Potatoes naturally take up cadmium. So you can actually get cadmium toxicity from potato. Rice takes up arsenic and cadmium and so on. So heavy metal, a change in the characteristic of heavy metal uptake could be a legitimate concern. Not for biotechnology, but for any breeding. I spoke to a guy who was doing some work on potatoes, and I said, well, I hope you're looking at potato glycoalkaloids. Oh, no, they're not biotech. I said, I don't give a toss. If you don't do that, there's a good chance, just by any manipulation, that you'll increase the glycoalkaloid levels there, because it's a response to all sorts of stressors and manipulations, and you can create toxic potatoes quite easily. But it's predictable, and it's easily checked. You just do a HPLC assay for potato glycoalkaloids. Right? So why wouldn't you do it? It's a one-day job or a two-day job after you set up your HPLC. And of course, in these various secondary metabolites, some of them are carcinogens. Aristolochic acid is a traditional Chinese herbal carcinogen, right? And Taiwan has the highest incidence of renal tumours in the world because they use a lot of herbal medicines that have aristolochic acid in it. After 20 years of as a toxicologist looking at human drugs, pesticides, it is the most carcinogenic and the most concerning carcinogen I've ever laid eyes on. One dose is enough to give you cancer some period later. A minuscule dose, repeatedly, it's the classic cumulative dose toxicity. Right? So it depends on the total dose, not how long you're taking it. It's really quite an astonishing carcinogen. So I worry more about carcinogens from the environment than I do from all the things that the popular press and so on get concerned with. Specific organ toxins, peroxide alkaloids. Some of these are really interesting. The mechanism that detoxifies peroxide alkaloids requires glutathione, and glutathione requires cysteine, which is a thiol amino acid. And in a poor diet with low animal proteins, you've got less of that. And so it's not surprising you see more pyrrolizine toxicity in those communities, particularly during those times when their diet's poor, right? Because they have less of that enzyme to detoxify. Okay, next slide. Again. All right, so secondary metabolites, the toxicity is not random, it's very targeted. There's a lot of energy and machinery invested in producing these things, so they're not just going to occur randomly, because otherwise you'd get rid of that if you didn't need it. So this is digitalis, the fox type. Quite a pretty little flower. I haven't got a colour photo there, but it produces digitoxin. Not only is it a specific cardiotoxin for mammals, and if a mammal eats that, it dies rather more quickly than the peroxide alkaloids. It's very toxic. But we use it as a human drug because it increases the strength of the heart beating at very, very small doses. But it also has this moiety here, which is, which gives it a surfactant-like property, which helps break down the cell walls of microorganisms that are attacking it. So that's being produced as a herbivore anti-feedant and an antimicrobial. It's a pesticide, very toxic to humans, but it's a pesticide that's naturally produced by this plant to protect it against major environmental threats. Next, please. All right, and I won't go through this in detail, but you can pick up plenty of graphs and text that discuss the protective nature of secondary metabolites. Okay? So, key take home is that secondary metabolites are there for a very specific purpose. They're not a random thing that's occurred, and they generally are derived from other physiological components that have been slightly altered or upregulated to produce these. And this is just an example of the effect of the environment and agronomic practices on the levels of secondary metabolites. Right? So this is sorolins, which are produced by celery. And if the sorolins are too high, it can cause quite a severe photodermatitis, which I'll show you in a moment. Right? And so all sorts of things can affect it in response to disease, heat, cold, light, air quality, insect damage, agricultural practices, how much water it gets, how much fertiliser it's got, what you sprayed it with to protect it, will all up and down regulate it. And we haven't even started talking about biotechnology now. This is all conventional, highly variable, up to 2 orders of magnitude. So in terms of this toxin, which naturally occurs in celery, if I was doing any change to that, conventional breeding, cell culture, biotech, I'd want to be looking at the level of the sorrelins and see whether that's up or down regulated. Because it exists already. Up or down regulating something that exists in the plant, existing machinery, is not as challenging as a de novo creation. So that's a legitimate issue. Next slide. All right. So crop composition and variability in it is a result of a range of these practices and environmental variables. The compositional variability due to environmental variation is greater than that secondary to gene insertion or alteration, other than the variation specifically expressed by the gene itself. Next. So this is just an example. These are all conventional wheat breeds, and this is 7 varieties of conventional wheat, 6 locations in the same year. And you can see there's huge variation in these different amino acids. And in any analyte you pick, this is just one example, you'll get this massive variation. Okay? So the idea that we're going to do all this compositional analysis on a GM crop, but not require it for any other form of breeding, it doesn't make a lot of sense, to be honest. Next slide. And again. Okay. These are all conventional soybeans. Right? Now, if I do a compositional analysis and compare that with that, they're going to be dramatically different, aren't they? Why? Because just the proportion of surface coating to the internal composition is going to be different because of the size difference, that's all. Right? So compositional variation can be huge for no reason other than the seed size or the grain size. It can be quite banal, really. And clearly there's nothing homogeneous about that. There's conventional crops of soybean, any of those could find their way into commerce. If you had enough of an increased number of pods for this compared to this, this might actually be a preferred economic crop. But of course, if you only have the same number of seeds, well then you probably prefer to grow that. And as you say, it's just due to the different proportions of the various layers and components of the seed as you change the size. Gene insertion, and we get some unintended changes, whatever they might be. In normal biotechnology, it's different for some things which are vegetatively propagated, but for sexually propagated crops, we're then going to cross back to the original parent. Every time you cross back, you're losing more and more of everything other than the particular trait you put in. So even if it were plausible that you could get some adverse change initially, it's less and less plausible as you go on, because you eventually end up with 99.2% of the parent line and less and less and less and less of anything other than the specific event that you put in there. Next one, please. All right, so natural food is a myth. That's a natural carrot. I'd rather eat that. That's natural lettuce, I'm saying. That's natural corn. I don't think anybody would want to eat that. I don't think you could bite through it, frankly. Next. There is more genetic variability between 2 ears of conventional corn than there is between humans and chimpanzees. We're worried about this one gene insertion into them. Okay, next slide please. And look, there are such huge variability in barley, conventional, all again. Tomatoes, right? These little things here which are all seed and skin. That's a natural tomato. You want to eat natural food, you can eat that if you like, but I think it'd be awful. You'd have to put it through a sieve before you could eat it. And these have all occurred through selection and breeding process, all conventional. Some of this has occurred by sticking a cobalt-60 source in the centre of a big field and massively irradiating these plants. Of course, we don't regulate that at all, which is again rather quaint. And again. Wheat is actually 2 completely different species that have crossbred to produce a polyploid variety, which gets up to hexaploid in some of the more recent varieties. Right? And that's whole chromosomes transferred across. don't seem to be too concerned about that. Next one, please. All right, mates. What do we know from nature? I mean, nature does a lot of experiments for us. We just got to go and get the data. I mean, that have been done for us. This has probably gone up. This slide's a couple of years old. There's more than 50 million identified single nucleotide polymorphisms, random mutations, across corn. Not once, not ever has any of that produced a toxic protein, for all the reasons I outlined earlier. If you take the same protein, the same functional protein, from any two corn lines of 300 to 400 amino acids, they'll differ on average by 3 to 4 amino acids, just due to SNPs. And what we're worried about, this one gene being placed in that genome creating a toxic protein. I mean, it's just not plausible. 85% of the genome sequence of the reference inbred line is identified as transposable elements, jumping genes. And jumping genes, they don't neatly insert themselves between two other genes, they just land anywhere. They can go from one chromosome to another. They can go from the mitochondria into the nuclear genome, right? And if they disturb a gene that's essential for the function of that plant, well, it doesn't grow, it dies, right? But it's never produced a toxic protein in corn, never. Yellow maize, so corn is supposed to be white, actually. Corn was always white in those days. But the yellow maize is a result of a particular insertion into the phytoene synthesase promoter region. Prevents the carotenoid pathway shutdown in the seed. This is an example of the activation of the dormant pathway. But the pathway is active and maintained and sustained and repaired and kept active in the rest of the plant. It's shut down by a specific change for the corn itself. Right? So because it's maintained active, all of the machinery and functionality is there, barring a shutdown mechanism in the plant. So that is an example where you could do it. So if you had something produced, let's say you're looking at zucchini or some other vegetable that produces a toxin in the leaf, and you're doing some sort of manipulation, it doesn't matter whether it's biotech or conventional, it would make sense to look for the toxin that's normally present in the leaf in the vegetable, because you could reactivate in the edible part a pathway that was dormant there but active elsewhere. But you're not going to create, you're not going to go back a million years in evolution. Right? It's just not going to happen. And if it could happen, well, of course, you would have done it dozens of times up here. Next one. Next. All right. Now, people have been buying seed from commercial producers forever. Why? Because they produce a nice homogeneous seed that's got the hybrid vigour. Right? It's true in animal husbandry as well. In Australia, we eat a lot of lamb, a lot of sheep. And when you want to produce sheep for meat, you breed two lines together, and it's the progeny from that you eat, because they grow bigger and they grow fatter and they put on more meat for any given amount of feed. Well, that's true also for your plants. You want to know you've got a predictable yield. Okay? And all these variations you see in these are different types of corn that can still be found in South America. It's just due to these SNPs and jumping genes and so on, altering existing pathways to produce different colours. Next slide, please. Other way. Yeah. Can you just go back, or maybe I'll listen? Yeah, go forward, sorry. Now, the same is true in soybeans. Now, the soybean people have been a bit lazy. They haven't been doing as much work as the maize people, and they've only found 5 million single nucleotide polymorphisms. But the count's going up rather rapidly. All right. And this is a pedigree. I mean, many of you will know more about these things than I do. Can you just push the button again? These numbers here in the sort of light pinky colour are the number of gene differences between related lines, and there's hundreds, right, between closely related lines. So what is true for maize is true for soy, and every plant anybody's ever looked at, the same thing is true. Right? Massive numbers of point mutations going on. Cool. Rice also has jumping genes. This is Gimbozo, which is an ancestor to modern rice varieties. On average, there's 49 to 63 new insertions per plant per generation. So recombinant DNA technology is going on a farmer's field, even if they're growing organic. Right? It's just happening all the time. The newer varieties they've reduced that. And why have they reduced that? To get predictability and consistency in crop. You buy a bag of seed from a producer, you want to know what's going to come out the end of the growing season, right? So they've made them more stable, but there's still one new insertion for every 3 plants per generation. Can you push forward again? So we get the pictures up. Alright, this is a traditional tomato, and you'll notice there's 4 lobes in a tomato. This is the ox heart, and you notice there's lots and lots of different lobes. That change is due to an insertion which causes the multiple lobe structure of the ox heart. So, existing machinery, altered, changes the structure, but it's still a tomato. Okay. Next. And as I say, jumping genes don't just come from one area of the nuclear genome to another. They can come from mitochondria. Each of these arrows show a gene that is inserted from the mitochondria into the genome of different lines. Each of these are different chromosomes of different conventional lines of corn, of maize. Okay? So there's a lot of recombinant DNA technology going on naturally in all the plants that we eat. Next slide, please. Same is true for, and again. Right, so the same is true for viruses. Retroviruses insert into the gene, so that's a different species inserting into the gene. Again, the layer, most of you are probably familiar with much of this. It's true in tomatoes, these are retroviruses inserted into the tomato genome. Again. The Green Revolution. Look how tall the wheat was when this painting was painted. I mean, they might have been really short people, but I don't think so. Right. So this is 1565, and again. So the big part of the Green Revolution that increased crop yields was simply to reduce the amount of energy and soil fertility going into producing all this useless stalk and producing a high yield by bringing down the size. And again. Right, that's due to a slip into transcription factors. So again, it's interfering with a pathway. So it's up or down regulating existing pathways in the plant. So as a regulator, one of my core issues in whatever's going on is, what is the plant that's being altered? What are you putting in it? What is it about this plant that could be up or down regulated, that we know about already, that could cause me concern? It won't be a mystery. We've been eating these things for so long, if any change could cause an up or down regulation, we'd know about it. So we know about it for potatoes, we know about it for zucchini or courgettes, depending where they come from. We know about it for cassava. Right? So we know the toxins that exist in these plants. So it's not a mystery to us, but it's a genuine, legitimate question as to whether you've up or down regulated an endogenous toxin. And that's something I would be always looking for. And it's not about biotech, it's about any breed. And again, presence-absence variation. There's massive differences between varieties in genes that are simply completely absent in one plant, one variety, and present in another variety. So that too is not uncommon. Next slide, please. And this is an ad, a legitimate ad out of the newspaper. In the old days, he'd take some seeds, stick them in a radiation field, mutate it sort of randomly, and they'd sell them to gardeners around the place. Just to, no idea. Can you just push it forward a couple of times? Right, completely unpredictable. Unlike anything, you just go back in. Oops, back in. Unlike anything yet known, and people would gaily buy this. Oh, I wonder what I'll get, Gee, the public attitudes have changed somewhat. All right, forward. And this is conventional breeding. This is a cobalt-60 source. All right, and so the strength of the radiation as mutagenesis decreases with the square of the distance from the source, so you get a nice, exponential drop. So out here, you're getting fairly low exposures, here high exposures. Massive genetic changes. Right? Completely unpredictable what you're going to get out of it. And essentially you just take the seeds, grow them and see what you get. If it looks good, you keep working with it. If it doesn't look good, you throw it away, or it doesn't grow, or whatever.
Is this basically done still? Is it?
There are thousands of varieties used by organic farmers, right? and other conventional farmers that have been derived this way. This facility still exists, but there are more sophisticated techniques, and it's fairly expensive in a sense. You've got all of this to maintain, and then you've got such random changes.
Right? We are not I don't know whether we're intervening now, but actually FAO has a joint program with the International Atomic Energy Agency that promotes capacity for the use of induced mutations. But I guess we can talk about this at the end of the seminar.
I mean, it's got the ridiculous point. I've done some work with CSIRO in Australia. That's familiar to. It's a scientific research organisation. Right? And they used biotechnology to produce the traits they wanted in a particular crop, right? Just so that they could make sure it worked and characterise it and work out which pathways changed. Then they went back and started again using conventional techniques, because it was cheaper to do that than meet all of the technology requirements and to deal with the public, concerns around biotech. I mean, how absurd is that? Exactly the same output, right? Just silly, really. And that's one of those unintended consequences, really, of disproportionate and unscientific regulatory burdens that we face. Next slide, please. Now, some crops can be toxic, right? A lot of crops produce cyanogenic glycosides, cassava. It's a classic. Almonds, even grapes, the grape seeds have cyanogenic glycosides, apples have cyanogenic glycosides, right? And that's specific to one part of the plant, but any form of manipulation, it is plausible that that capacity to produce that toxin could move across the edible tissues. And I think you'd be silly in any form of crop development, conventional or otherwise, not to look at those endogenous toxins. But there's no regulatory requirement at the moment. Squash and zucchini, that's an example of selection over long periods of time by conventional means, selecting for plants that don't produce the toxin in the edible portion of the plant, but does in the leaves. And that stops pests eating the leaves, which is a good thing. But there are times when that change can revert because the pathway is active in the rest of the plant. It's just been made dormant in the edible part, so it's easy to reactivate. And there's a bunch of other things, such as the celery, the furanocoumarins, which cause contact photodermatitis, can be upregulated by all sorts of things: late harvest, pest pressure, altered agronomics. Can you push forward again? And again. And this is the furanocoumarins, celery, and again. Right, that's photocontact dermatitis, right? It's not a trivial thing. If I'd been working in my veggie garden and I ended up looking like that, I'd be pretty upset, right? And this sort of thing has occurred in people working in supermarkets, working with celery. Alright, so of millions of conventional varieties, only 2 have been reported to have unintended effects from toxins. So unintended, unexpected dermatitis and stomach aches. All involved elevated levels of known toxins, part of the OECD known plant toxins. And yes, we want to test for those. What about unknown toxins? In all the history of breeding, a toxin that did not exist at the genus level has never been reported to spontaneously arise through any breeding technique, no matter how much you alter the genome. Protein toxins, secondary metabolites, same for all. There's one report in an EFSA report where they claimed, this paper claimed, that they found a toxin in a sort of potato that had never been seen. And again. Alright, so this is a technique where you can compare the genome of 2 varieties. Right? So this is one that's being subjected to fast neutron bombardment, and it's basically these dots here. This is all just random noise, these dots up here. And when you get a little bit like this, it just shows you've knocked out a large chunk of the genome. Next. Okay, and again. These are 2 related conventional lines, massive genetic variation, and again. And this is a GM compared to its neo-isogenic line, and you can't see anything, it's just random noise. And that's totally what you'd expect, right? Because not only have you crossed it, then you've got all the back crossing, right, against, so you're back crossing this against this, and you keep doing it, and then basically you're comparing the same thing against the same thing, so you wouldn't expect to see anything. Well, if this isn't causing anybody to get sick, what's the chances of this ever producing something you're going to worry about? Next one, please. Alright, so we can answer our original questions. How variable is a phenotype? Vary due to a whole pile of common things that go on. How common are gene assertions? Very common. What are the consequences? Never produces de novo toxicity, but most certainly can up or down regulate existing pathways. No question about that, and that can be a human health risk. And we need to be awake to that. How common are point mutations? Very common, exceptionally common. But again, never produce toxicity. Is there evidence for activation of dormant pathways? Yes. If they are existing in one part of the plant and dormant specifically in another part, yes, you can regenerate or reactivate that pathway because it's maintained. The DNA is conserved and the functionality is conserved. Is there any evidence for going back in the genetic evolutionary history? No. Never, despite all of the massive changes. Next slide, please. Just okay. So, crops are genetically unstable. We know that. And a little bit of manipulation with them isn't changing much. We don't get any toxicity from any of that. So from a pure scientific perspective, the need to regulate NBTs in any biotechnology should be weighed against the now substantial body of evidence of highly plastic plant genomes and the absence of consequences of hazards beyond the up-regulation of production of known toxins. So what should we do for regulation? So that you can close it down now. Basically finished. What should we do for regulation? I can tell you with 100% assurance what the consequences of putting a specific gene into a specific plant is if you can characterise that plant. Does it produce any toxin in nature, ever? Right? And is there any possibility of that being conserved, upregulated, downregulated, whatever? So that's the first question you'd ask. And then the second question you ask is, what is the function of this gene? Is it clean? Right? Is it very precise? Is it clean? Where does it come from? Because you might, maybe carry something over. If it's clean, you're assessing what that gene codes for, specifically, and the plant that it's going into. So in terms of regulation, as a regulator, I'd be quite happy to have a well-characterised gene insert and a well-characterised plant. And what I'm suggesting to a number of countries around the world is the next step in regulation should be, we should say, we know a lot about maize. We know a hell of a lot about soybeans and canola. We should approve maize and canola and soybeans for genetic modification, just completely. Because they know they don't produce anything we don't worry about. Then you look at the gene inserts, and you look very precisely at Cry1A or whatever it's coding for. And say, well, okay, we've done that a lot, we know a lot about that, we know a lot about its safety. So we'll approve the specific inserts and specific structures to go into any one of those. Okay? And you'll have 100% confidence in the outcome. Europe has spent $500 million on animal testing of genes. Completely and utterly worthless. Totally predictable from basic biology and what I've been talking about today. The only thing they'll ever get out of it is random noise. Right? I wrote a paper recently with Ilse on exactly that topic a couple of years ago now. And if you're let's say you're an analytical chemist, and I said to you, I want you to test the sewage sludge for toxins. It's a complex mixture. You must use a HPLC. You must use a C18 column, and you will use flame ionisation photometry as the detector. Nothing else will do. That's what you've got to do. And I want you to tell me if there are toxins in there. How can you do that? It's ridiculous. And that's exactly what they're doing in the rat study. You don't know whether the rat is sensitive. You don't know whether the toxin is there or it's not there. You don't know whether the rat will actually respond. You don't know what the dose is. Okay? You can't do a calibration curve. So normally in toxicology you do a series of doses to make sure you got to a dose range that the rat was sensitive to avoid specific sensitivity issues between rats and humans. Well, if you can't do that because you can't get a dose range because you don't know what you're testing, right? And a whole bunch of other issues. So that's getting off topic a little bit. So I think there is an opportunity to help some of the smaller countries who are desperately trying to build their own indigenous biotechnology to deal with very specific issues for their people in remote areas who are often some of the poorest and most disadvantaged in the world. What rich Westerners do in their own country really isn't relevant except the attitudinal effect that it might have on those countries. But I think it'd be really useful for an organisation like FAO to promote something like an expert consultation on what the regulatory regime should now look like, given what we now know. Because frankly, it's moved on so far over the last 15 years. And people are dying and are being hurt and are being sick because of the disproportionate regulation. So it's a very good example of the precautionary principle actually hurting people. So that's my perspective. I'm happy to discuss and defend.
Well, thank you very much, Andrew. This is very knowledge intensive. I'm sure that you're going to generate a lot of questions and debate and comment. The floor is open for questions, comment. Okay, let's start from, I was first, okay, let's go this way, all right? You and then we can go like this.
Okay, thank you very much, Bartholomaeus, for the presentation. I mean, the passion that you have for the subject is quite obvious and, if you may, even infectious. And I just wish that those of us from FAO sitting around the table have that luxury to express our own personal interest in subject matters such as this. Biotechnology and its applications, especially in food and agriculture, will always remain a contentious issue. And science and its data, can always be used any way that they want. But something that, if you may, I would want us to kick off this conversation. It is the seeming comparison between genetic modification and spontaneous and or induced mutations. You made quite some allusions to the use of induced mutations to generate crop varieties, but I don't know how many of us understand that genetic modification involves the insertion of extraneous material, genetic material, that begins to express in the recipient genome. Spontaneous mutation or induced mutation does not introduce anything from outside. It only modifies things that you may, given sufficient space and time in nature, would occur. Could you clarify this?
Well, if you expose a plant to massive radiation, you're producing genes that have never existed in nature ever, in any species. Right? You aren't just modifying what's there, because let's face it, what is a gene? It's a series of nucleotides, right? Same nucleotides in every species. So the only thing that's different between whether it's induced or comes from external is that sequence, and you can produce that sequence either by massive radiation or by bringing the whole gene in, and it's actually more precise. At least you have a gene that is existing in nature and you know what it does, whereas what you're creating through mutation is a gene that has never existed in nature, in any species, at any time in all of evolutionary history, possibly. Right? You've got no idea what it's doing, and in fact, a lot of the varieties that have been produced by radiation, nobody actually knows the specifics of the gene changes that were introduced. And nobody's got a clue about the additional genetic changes that aren't related to the specific trait that they are actually selecting for. So I would argue that you are getting closer to nature and more precise and less unintended consequences by inserting a specific gene, or with the new technologies you can do gene editing. Right? And all that is just changing the sequence of the nucleotides.
Well, Mr. Chair, this will be my last intervention on this, but what you have said is not exactly factually right. There are techniques, very simple ones, for characterizing the modifications to genes. And you had alluded to single nucleotide polymorphisms. Most of the useful mutations that you find in crops are no more than indels or changing one nucleotide to the other form of it. Like the green revolution dwarf genes, for instance, just a single nucleotide change in the particular gene. And it is also not right to say that induced mutations are producing genes that never occurred in nature. That is just not possible. Given sufficient time and space, yes, adenine can always be changed to thymine. It is just a matter of, do you have the time to concentrate all that and into a variety? So this is not to say that I have anything against genetic modification. It's also not to say that FAO has anything against genetic modification. actually, we are going to have a symposium on agricultural biotechnologies early next year. And the idea is FAO provides this neutral platform for people with all shades of opinion to come and debate. And what do? We avail our member countries of the opportunity to access the facts and make evidence-based decisions. But to begin to then, compare and even negatively, the inserting of foreign materials into a genome and, something that would be like a natural process. If there was no mutation, you wouldn't have agriculture the way we have it today. Almond will still be bitter. The pods of peas will still be shattering. So, this is just the little contribution I want to make. We stick to the facts and make our case, and it makes us a lot more credible.
So I would argue that it's wrong to say, it's not correct to say you're inserting foreign material, because it's the same nucleotide, just a different sequence. Right? And the smaller the sequence you look at, the more likely it is to already exist in that plant. And if you take a plant and you stick it in the radiation field, you're not producing one SNP or two SNPs, you're producing massive. That example I showed where they compared them, and you lost a 6 kilobyte block. Right? That's the sort of thing that goes on with mutation. And in nature, you don't get those sort of massive changes in one go, and then they are lost over a period of time because they're not conserved. There's no reason to conserve. It's not functional. Okay? So the fact that a gene exists, you've taken from one plant and put into another, is no different to crossing two species and taking whole chromosomes that don't exist in nature. If you take it even from a fish, the degree to which the enzymes in plants and animals is conserved evolutionary, once something has been evolved that works, it tends to be highly conserved across a broad range of species. Right? The fact that it doesn't exist in that species, but it exists in 100,000 others, right, that doesn't make it unnatural to be in there. It just means it's easier to do it that way than another way. And you can use other techniques to create exactly the same outcome that are then considered conventional and not regulated. I think the big problem for me is there is a massive inconsistency that has nothing to do with safety and has everything to do with philosophy, let's kind of call it that. And there's massive commercial interests. Now, I don't care if Monsanto and Dow go broke tomorrow. That's not my concern. There are other ways of dealing with the things they deal with. But gee, I really do care if a small university in Indonesia can't help a community that conventional breeding hasn't been able to help, who would be tremendously improved in terms of their quality of life and food security and exposure to pesticides and fungicides just by producing a potato that wasn't susceptible to lake blight. So, it's time to rethink, regardless of how you define the technical detail. I think we need to get above the technical detail and say, what do we know now? It's safe. We know it's safe, provided you've got people doing it who know what they're doing. It's certainly no more dangerous than conventional breeding. Do we really want to block technologies that have huge humanitarian benefits because of a philosophical objection of rich countries who can choose to do otherwise? I think it's actually
important. Let's actually move to, yes.
My name is Sebastian Hjelm. I work for the Soviet Secretary. And I was actually about to start with that point that you made last about, in my opinion, this is not really about food safety at all. I mean, it's been a political issue for a long time, and I think the way that we have built food standards and food regulations over the world and based them on food safety and making it the only legitimate concern, it just comes back at us now. We can't have the socio-economic discussion about whether we want to have Monsanto, whether we want to have big profits in agriculture for this technology. So that's why we are discussing safety. I fully agree with everything that you said, and I also agree with that we should make this technology available to those who want to use it. And right now we're doing a really bad job at that. But since the only argument that is given to those who oppose GMOs of mostly political reasons, that's why we're having this discussion. And it's a kind of a non-discussion, has been for a long time. Then I actually have two questions as well. You said reactivation of dormant genes that are already in there in the plant. And you gave the zucchini example. I was just wondering about if you could elaborate a bit on sort of how would this happen? Since the way I understand biotechnology is that we take a gene which is rather specific and we put it into the plant in a certain place. And we don't use any random radiation or stuff like that. So what is, I mean, how does it awaken that dormant gene, which is obviously functioning for the leaves very well, and how does it actually activate it in the fruit? My other question is that you spoke of biotechnology as something which, you know, when we look at maize varieties and wheat varieties and stuff, but obviously there's people out there who are willing to play with much more fantastical combinations of genes. So do you see any worry in, let's say, combining human genes with whatever, fish genes, and making sort of funny animals and sort of, or funny plants producing the most wondrous proteins? Could there be a risk in that?
Okay, so I'll address three issues actually within that. I'm not a plant geneticist, so I won't go into the specifics, but in terms of reactivating a pathway that's dormant in a specific tissue, any conventional or biotechnology manipulation of the genes will require use of a number of techniques, including cell culture and backcrossing and all these things. They alone, without any insertion of a gene, are capable of upregulating or downregulating endogenous pathways through the introduction of small genetic changes that just occur through the manipulation in the process. So it's not specific to biotechnology. And so as a generic term, whether it's conventionally produced, I actually do believe that, in a diverse sort of sense, the Canadian approach where all manipulation of genetic material, whether conventional or otherwise in plants, is technically covered by regulation. Because I can see a number of circumstances where a whole range of conventional manipulations of plants that have an endogenous pathway for toxicity should at the least have a mandatory requirement for testing what's happened in those endogenous pathways. The other thing I would say is the reason we're not getting very far in convincing the public of this is because we're not paying attention to what we know about the art of rhetoric, persuasive argument, which goes right back to the ancient Greeks. We keep focusing on the logos, the science. Right? So Plato would say that there are three components to an effective argument: logos, pathos, and ethos. Ethos is who is talking to you. What's their credibility? What have they got in it? What might their attitudes and background be? What's their conflict of interest? Pathos, what do you feel about this stuff? What worries you? Taking a human gene and sticking it into a sheep. A lot of people out there will say, "Oh, how can I be eating a person?" Well, actually, there's a lot of genes in sheep that exist in humans too. We all have a gene for a tail. It just doesn't get expressed in humans. Right? The genome of a rat is actually very, very close to the genome of a human. Right? So, from a purely scientific perspective, you can say, well, so what? It's just a sequence of amino acids that are being expressed. But we're failing to address the other issues that affect people, and that is the pathos. Why should I care? Now, I've had this same debate, so because I come from human pharmaceutical side, I've had to talk to people who are against vaccines. Oh, vaccines cause autism. No, it doesn't. Do I convince anybody? I can say we've done clinical trials, it doesn't do it. Do I convince anybody? In fact, when I was with Fazan's, we actually did a study where we went out, this is about nanotechnology at the time, and we got a group of people in a room, and we surveyed them and said, what do you think about this? And there'll be the group up here which says, oh, it's great, wonderful applications, really, think it's fantastic. And there'll be a group down here, and it's manipulating nature and it's dangerous and we don't know what's going to do, and the power rests with the big corporations and I wasn't consulted, right? And then there's a bunch in the middle. And then you hit them with information. You educate them. Does anybody move from here to here? No. Maybe one or two, and maybe one or two move from here to here. What mostly happens is the people in the middle split. If you do a really good job, more will go to the positive than the negative, but there'll always be a group who disagree. Now, if you overlay that with the multi-billion-dollar industrial issues around crop breeding, organics is a huge industry today. And the more you can make people worry about the other food in the supermarket, the more they're going to buy it, right? And of course, on the other side, you've got huge companies making mega profits selling Roundup Ready canola or whatever it is. And so there's this huge perceived conflict of interest. And so you've got this, dimorphism. So in terms of getting people to vaccinate their kids, do you just keep banging on about how safe it is? You show them some pictures of children, the age of their children, who are dying from measles. What are you attacking? You're attacking the pathos. You care about your kid, you want to see them like this, right? That actually has more sway. But as a regulator, of course, you never have any scope for that. You have to stick to the facts. Politicians do. And I think that's the problem. One of the good things about the nanotech consultation that FAO ran is they had a group of communicators as a separate part of the discussion. How do we communicate this to the general public, and to provide advice for nation states on how to better inform their community? And a lot of that was about dealing with the positives, the humanitarian benefits. Yellow rice, cassava that expresses Bt but has a low level of cytogenic glycosides. Why is that important? Because when the crop's in a bad year, they're producing more cassava, and guess what? They need to eat the cassava more because they're running out of food, but there's more cytogenic glycosides, so they get toxicity. Put Bt in there, you can drop the level of cytogenic glycosides. It's still resistant to the pests, because if you take the cytogenic glycosides out without putting the Bt in there, it just gets eaten by the pests. Huge benefit. And they don't need to put all these pesticides on. I did an assessment of a pesticide some years ago in Ceylon. And the idea there, their concept of occupational health and safety was a pair of thongs. Because that's all they wore, a pair of thongs and a pair of shorts, naked top, naked legs, and a backpack on spraying. That's occupational health and safety. If you can stop people using those pesticides, you'll do more for the public health and safety of those workers than almost anything else I can think of.
Okay, let's take the last 1, 2, 3 questions. Please write them down, and then we're going to take them all. Yes.
Christine Fu from Rotterdam Convention Secretariat. I've done also pesticide risk assessment in the past. Thank you very much for your very thought-provoking presentation. And I would like to say I fully agree with your statement about natural. I also would prefer taking an aspirin compared to chewing on a willow branch. However, natural, the term is not defined as, for example, is organic. At least I know that from the European context, you have a legal definition on what can be called organic and what not. But that just, I have actually a lot of questions which I don't want to ask you all here, because I would like to give the chance also to others. These questions I can ask you, but I don't expect an answer. These questions would include: what are the quantified advantages of genetically modified plants compared to the disadvantages? And I'm talking not just about the scientific part of it. Scientifically, as I said, I did pesticide risk assessment, and I did mainly the scientific side. I didn't look at the economical and the social side. But you were giving the example of parents looking at children who die because they haven't been vaccinated. Maybe one could also look at farmers who get into an enormous dependency from industry if they are supposed to just grow genetically modified plants. This is just one thing. The other thing I was asking myself is there nothing else than conventional radiation breeding compared to genetically modified organism breeding? Isn't there something else?
There's many. I just use that as an extreme example of, this
so
is
we were talking only about the extremes. So we were basically comparing the devil with a beast, or whatever you would call it. And I don't expect an answer to that. What I would be really interested in, you were saying that you are looking at those countries you mentioned in the beginning, and whether universities in those countries couldn't help the small farmers, as I understand, the family farmers in those countries, to come up with varieties, potatoes that are not affected by blight. Now, how would that look like in practice? Would that also create a sort of dependency, then not from big industry, but from university, from science, from whomsoever? Could these smallholders do that themselves, or would they again depend on some other institutions? That is the one question I select where I would really hope for an answer.
Okay.
Thank you very much.
Okay, let's hear from others. Yes.
Professor, I would like to thank you. I'm Diogo Machado, I'm an agronomist. I've been interested in this theme for all my life. I think your presentation was very good. I agree with your points. And I thank you for leaving us a challenge so that the FAO can promote an expert consultation as to what the regulatory regime should look like. I think that's a good way forward. In Europe, we've come to this situation that we have the European Food Security Agency approving or saying that the new varieties should be approved, but politically the European Commission does not want to take the onus of responsibility anymore, so it's passing it to each member state. This has to do with the way Europe takes decisions now following the Treaty of Lisbon. That's just made it all more complicated. I won't go into that. But I see consequences. Consequences for the European industry. There's a shortage of protein. It's very hard to import soybeans. But also, and maybe more related to the work we do, how that reflects in developing countries, and especially in Africa, where there isn't conventional technology and where the use of genetically modified organisms and the technologies associated with it would make agriculture much easier, you know, technically. And so I guess that's where my concern really lies. This is just some points to reflect upon and to thank you for your presentation. Thank you very much.
Okay, Sam.
Okay, thanks again also for very interesting presentation and for provoking this kind of discussion. Mine is actually a very basic one. I appreciate the point about the similarities of genome between different species, but the basic question is about, for me, is about gene transfer. If humans were to eat plants with altered genes, has there been any proven case of introducing a new DNA sort of into the genome of somebody eating such plants? And if so, how could that be detected and really verified?
Okay. So let me.
No, there's the
sorry,
last
one more. Sorry.
Yes, a last short on technical. As far as I know, there's no direct test to check for pleiotropic effects, and I would like to know whether there's an update on that. What do you think about these effects?
So let's quickly try and cover most of those in.
You have 5 minutes.
You do like to set a challenge, don't you? Okay. The situation with farmers and GMOs is no different to non-GMOs. If I have a patented seed, you have exactly the same obligations legally as if it's a GM or a non-GM. Okay? Conventional bred or not, so it's not different. Why do farmers grow GMs? It makes more money. Why does it make more money? Less pesticide input, less is actually genuinely true. Less pesticide input, less pest damage, higher yields. They don't have to grow them. They don't sign a 20-year contract with Monsanto that they only will grow their seed. They can change any year they like. If a farmer wants to keep seed back after a GM, he's grown a GM crop, he's allowed to. But he's not allowed to select back to get the identical gene composition of what he bought, because that's a patented seed. And that's true whether it's conventional or non-conventional. Okay? In terms of golden rice and the potatoes, the idea is that it's developed and you just give it to the farmers and they do what they need to do. And if they can keep it a reasonable productivity, that's fine. But the reason people buy seed from seed producers, as I said earlier, is because the hybrid has the hybrid vigor, and if you buy it from a seed producer, you get a higher yield. So you pay for your seed, yes, but you get a return in that you get a higher yield. And that's the only reason people buy seed, otherwise nobody would do it. Okay?
So, just a short insertion, sorry, Robert. We would I would expect that if we would allow GM seeds to be developed far more than now, we would probably see non-profit organizations. Is there any indication of this that governments or other do-gooders would do the breeding but give the seeds more or less for free? Not wanting the billion-dollar profit that Monsanto is seeking.
Yeah. Look, the problem with Monsanto, they'll only deal with crops that are broad acre crops because that's where the money is. But there are more crop varieties being developed in universities and by non-government organisations than all of the seed-producing companies combined. Bill Gates Foundation is one that's developing some, the Warren Buffett Foundation is doing some, and lots of universities are doing it. But they're all sitting on, not those big ones, but there are thousands and thousands of varieties developed for communities in South America and remote locations sitting on shelves in universities because nobody's got $10 million to do useless studies, completely worthless studies, to demonstrate safety that we already know is there. Specific example on that aspect. In the US produces 9 billion animals a year for food. 90% of them are fed GMOs. If there was any toxicity, what is the most sensitive endpoint in toxicity that you'd see in a herd? Reduced fertility and reduced weight gain per kilo of food given, right? So if these GMOs are producing toxicity and you've got 9 billion animals, right, no farmer would ever feed their animal GMOs, would you? Because they'd be losing money. They wouldn't be able to produce the next generation of their disease. So the toxicity issue is, you can pick any endpoint you like, hypothesise pleiotropic effect, anything you like. It's not happening. If you want to talk about DNA being taken up, you know, we've got jumping genes going on all over the place. We've got massive mutations and we're not taking those genes up. And so, yes, small components of RNA and DNA can be taken up. And I've got another talk, I won't get into it now, I've only got 2 minutes, on RNAis and so on. And again, there is no evidence of RNAis being active in humans. Some specific insects, yes, and you can target those specifically with RNAis, but not generally. Economics versus specific safety, well, I think I addressed that, and dependency. Really, I think the furphies. You don't have to continue growing this stuff. If it doesn't produce more money for you, if it doesn't produce more money by a bigger crop and less inputs. And the big thing in agriculture is the cost of pesticides. And safety. If you can reduce the amount of pesticides you apply because you're producing BT in the plant, less cost to produce it, greater yield, less concern about pest pressure. And if you can combine that in a stack with drought tolerance and whatever other environmental concern you have, then for the farmer, they're healthier, they pay less to produce the crop, and they can earn more money when they sell. And that's the reality. There's a lot, you can get on the internet and you can support any position you want to support with cherry-picking data. But there is a stronger consensus about the safety and value of GM foods than there is amongst scientists who really understand it, than there is around global warming. And yet, we've got massive worldwide response to global warming, which is legitimate and important, and we just ignore the fact that communities around the world are suffering because of a disproportionate regulatory regime around GMs, and I think it's unethical, frankly. And I do all this for nothing. I'm a consultant. Right? I can earn more in a day than most people earn in a week, but I do these sorts of talks all around the world for nothing because I just feel that, having retired from 20, 30 years, it's an issue that I care enough about to want to spend my time, trying to make a little bit of a difference in terms of attitudes that are causing people harm. I'll stop.
Okay, I think, thank you again. We were able to finish exactly on time, and I think this is what were given us time for this seminar. And as I said at the beginning, this topic is a topic that's going to generate debate and discussion for a long time to come. There are such issues in the world that need time to be resolved, need time for people to fully understand, because there's science behind, and this science you have to understand. And most of our politicians, they are not scientists, and they need to understand from scientists. And I think scientists are getting there, but they have not been very efficient in communicating, to our decision makers about what really GM is, what are the perceived or potential risks that we have, and how we can actually address the food safety issues. And this is, people are kind of reserved because of these issues they don't understand really clearly. So I think people like you and other colleagues have to get there, and maybe we have to get people from communication to transform them into scientists and get back to communicate that to our decision makers. We are also pleased that, as Bas said, that next year we're going to have a symposium here at FAO, you know, and I'm part of the task force, but you can see that a lot of let's say knowledgeable people are coming from the world to debate this issue, because policy has to be based on fact, on scientific evidence. This is sometimes lacking, and I think we hope that this symposium will provide, the answer to those things. So thank you for the organizers, and to the network, and thank you for coming. I think you are still around. So Christine has a lot of questions. You can still continue in the corridor.
Oh, we're going to have lunch upstairs. My family's here. So come up
so
and
with
join.
that, let's give a big hand to Andrew.